just escape out of here. And it goes right there.
She's already last.
You wanna see if they
Do you have
see.
slides for Merikangas?
Nothing for Kathleen, Joe, yes,
Yes.
Amy,
Okay.
yes.
All right. You can put the walk-in slides back up.
Okay.
The next speaker checked hers as well. She came.
Kathleen is local, and she's not here yet.
Oh, gotcha.
And I'm thinking, so they're closing down roads for like the women's march and stuff. So I'm about to call her. I just wanted to make sure she didn't sneak in. But if she's already last. Yeah, I can take them. I'll just making sure she's already last.
Welcome, everybody. Good morning. You're all very welcome. Please take a seat. Thank you. I'm Sandra Kooij. I'm a psychiatrist from the Netherlands, and I feel honored to chair this very interesting session on the interaction between genes, environment, and health and comorbidity. And we have three speakers, and we will divide the time between them, honestly. But the third one has not yet arrived, unfortunately, probably due to traffic jam in the city. So we expect her to arrive maybe a bit later. But for the time being, we agreed among the speakers to give them a little bit more time. She got there. Oh, okay, no problem, she arrived. So every speaker will have 30 minutes and 10 minutes for discussion with you, the audience. So if you have a question, please take notes and be prepared to ask them to the speakers after their talk. The first speaker is Professor Amy Arnsten from Yale University. She's a professor of neuroscience and an expert on the prefrontal cortex. That's the target of ADHD science in general. So we're delighted that she's here to brief us on her latest research. Amy Arnsten. Sandra, I'm not sure the slides are on. Do you know how to get them? Please, can we get some help to get the slides in place?
Oh, there it is.
Yeah. So, thank you.
Well, thank you all, especially Jeff for the original invitation to speak with you all today. And I wanted to start with my disclosures because for once they're actually relevant. So my lab developed Intuniv, and Yale and I receive royalties from Shire for the US sales of Intuniv, not from any generic or international sales. And since it's generic, these royalties are actually almost zero at this point anyway, but legally I have to say that. I also consult with Lundbeck and Blackthorn, trying to develop therapeutics based on what we know about prefrontal cortex. So let's start with the symptoms of ADHD that all of you know better than I do, the impaired regulation of attention, impulse control, and that it is evident at an early age and often continues into adulthood, and is often comorbid with many disorders, for example, oppositional defiant disorder, which is inappropriate aggression, Tourette's, inappropriate movements as tics, and most relevant for today's session, ADHD can mimic other disorders and lead to impaired diagnosis. For example, stress or trauma, PTSD, a family going through a divorce. A general physician might not be able to know the difference between the impaired attention due to trauma versus that with ADHD, and often why they need you guys in this room to help them with that. Same thing with childhood bipolar disorder, and something that we'll focus on today, lead poisoning. All of these involve dysfunction of the prefrontal cortex, especially in the right hemisphere, and so that will be the focus of my talk today. So some basics on prefrontal cortex, its function and topography. And I want to say that Dr. Helen Mayberg and I just made a PowerPoint on this, prefrontal and mental disorders, found at this YouTube link. And I saw it's not active right now. Yale must have deactivated it. But we're going to put it on the APSARD website. And if anybody wants the link, please feel free to email me, because the whole point of it is to educate physicians, med students on this important topic of the neurobiological basis of neuropsychiatric disorders. So the prefrontal cortex has many fundamental functions. It is one of our most newly evolved brain regions, and it has this unique ability to generate mental representations, to allow us to think about things that aren't actually happening, and to use those to thoughtfully guide our behavior, attention, and emotion. It is the foundation of abstract thought, and it's often tested in the lab, especially in animals, in working memory tasks. Am I getting too much of a... with the 2 mics, is it okay? Let me know if it's distracting. So prefrontal cortex does top-down control of attention, action, and emotion, representing a goal to inhibit inappropriate actions. And it allows us to plan ahead for the future, including the very near future, and to have the patience to wait for a larger reward instead of grabbing a smaller reward immediately, part of impulse control. Many of these things are part of what we often put under the umbrella of executive functions: planning and organizing, high-order decision-making, insight and judgment, and inhibition of inappropriate actions. And so the connections of prefrontal cortex allow us to see a topographic organization in how it carries out these top-down regulation of thought, action, and emotion. This is in a dorsal and lateral versus ventral and medial topography. So the regulation of thought and attention is more dorsolateral, with top-down control back to the association cortices, hippocampus, caudate, and down to brainstem. And this is based on stimulus relevance, so studying something boring for a test, and is opposite to bottom-up attention, where stimuli are processed based on whether they are salient, moving, bold, loud, such as video games. So prefrontal allows us to inhibit this and instead attend based on relevance. There are also circuits for regulating action, and in particular, inhibition of inappropriate impulsive actions we'll see is for many people on the right hemisphere. And this includes connections from this right inferior prefrontal to the motor and premotor cortex, including the supplementary motor area, and there are parts of the anterior cingulate that do this as well, that can be seen in functional imaging and also in studies in monkeys. And these circuits project down to the caudate, putamen, and subthalamic nucleus for either thoughtful inhibition, if we have time, or emergency stopping through the subthalamic nucleus if we have to do it immediately. And then we have on the ventral and medial surface the regulation of emotion, connections with the nucleus accumbens, hypothalamus, amygdala, and brainstem. And much of the output is from these very ventromedial areas, and also some from the anterior cingulate down to these limbic structures to regulate emotion. And this can be seen in these parallel loops through the basal ganglia and the cerebellum for the control of emotion, cognition, and action. Now, much of this work has been actually developed by studying the monkey cortex, and a simple mnemonic that Price came up with is that the dorsal and lateral parts of prefrontal represent the external world. They're getting inputs from visual, auditory, somatosensory systems, whereas our ventral and medial are representing our internal state. And this is where we have taste, olfaction, viscera, pain, and then our outputs to our viscera. So this can be a simpler way of thinking about it that I often find helpful. And of course, these areas strongly interconnect. For example, for dorsolateral being able to control emotion, and vice versa for emotion to color action. There is also a caudal to rostral topography, where caudally we have simple representations. Where did I leave my car this time? And then, I know that's not simple for some of us, and as we go increasingly rostral, it gets increasingly abstract. Representations of representations of representations. So our metacognitive abilities are up here in our frontal pole. And this is where we have things like insight. If you have lesions here, you have sociopathy, which is an important thing to say in Washington, D.C. I have bets about exactly where lesions are in certain people here. So there, in humans, also hemispheric specialization, where the left is generative and language generation in most people, and lesions produce depression, whereas the right hemisphere is often specialized for inhibition, and lesions induce impulsive manic and sociopathic symptoms. And as we said, the right inferior, especially for inhibiting inappropriate movements. And of course, this will be very relevant to ADHD. So let's look at, very briefly, prefrontal alterations in ADHD. And a great deal of this work has been done by Judy Rappaport's group at NIMH, where they look at thousands of typically developing kids versus those with various disorders. And I love this study by Phil Shaw, where he sees that in typically developing children, the left inferior prefrontal cortex at age 4 starts out larger than the right side. And then as we grow older, this is 20 years old, the right side slowly grows larger and larger until it's the larger one of the two. And this is what we call emotional maturity, right? And you can actually see it in their imaging studies. Well, in ADHD, this doesn't happen. The left starts out bigger, but it does not switch over to the right. In other words, the right inferior prefrontal is not showing the normal enlargement due to greater connections that would normally happen during typical development. And this right inferior prefrontal, both underactivity, reduced volume, has been seen for years, for example, by Katia Rubia et al., reduced prefrontal connectivity, volume. Many labs have seen these, often in different areas, but when you actually have people doing a task where they have to inhibit an impulsive motor act, this area often does not activate as it should. And Katia Rubia has shown that you can see patterns related to disorders. So this is ADHD, but in conduct disorder, it's more ventral, these more failure to control emotion. So fitting with what we know from monkey about the connections. So now that I've told you about that, let me dive into a little bit of our monkey work and the cellular basis of prefrontal functions. So prefrontal neurons generate and maintain goals for action, including being able to, the goal of inhibiting something inappropriate. And we can record from these cells in monkeys performing cognitive tasks. And in particular, this is the task that many of us use, developed by Pat Goldman-Rakish, who was at Yale but tragically died in 2003. So in this task, the monkey focuses on a central fixation spot to start a trial. A cue briefly flashes in one of these 8 locations. And then the cue disappears, and the monkey has to keep fixating and remember that location for 2, 5, 10 seconds. And then at the end of that delay period, the fixation point goes off, and that's the signal for the monkey to move its eyes to the remembered location. And if they're correct, they get a bit of their favorite juice reward. And then they can start the next trial by fixating again. But now the cue has changed, and they have to update their contents of working memory. Where did I park my car this time? And since my parking garage has 10 floors, I really relate to this task. So when we record from prefrontal, we find these cells called delay cells that can fire all the way across the delay. Each one of these is a trial where the cue had been here, and they can keep themselves firing that whole time, so at the end of the delay, they say, okay, look to the left. And these neurons have what's called a preferred direction, so they'll fire only if the cue had been here, not if it had been any of those other locations. And also importantly, they can keep firing even in the presence of distractors, and that's different from any other neuron in non-prefrontal areas. And this delay cell activity is also what's used to inhibit an inappropriate response. Pat showed that in 1993. So we've been able to see, and based on her work, that how a cell is able to do this is through a circuit, a microcircuit of pyramidal cells, they're shaped like pyramids, exciting each other to keep the information in mind. And they're also able to do this when we're, for example, looking away from a cue. So here we see them exciting each other to keep this going across the whole delay period. And the idea is the more of this you have, the longer and more strongly you can remember something. So we've been studying these circuits, how they do it, and we see it's through glutamate NMDA receptor synapses, where this cell can excite this one to keep information going. And these newly evolved circuits have on them potassium channels, and these can be opened by something called calcium cyclic AMP signaling. And when that happens, the information, the excitation is shunted away, and this cell stops firing. So it's a very powerful mechanism that keeps us from having seizures, but also this gets used by the arousal systems, including norepinephrine and dopamine, to coordinate our cognitive state with our arousal state. And what we see is this inverted U dose response between catecholamine levels, arousal levels, and cognitive function, where either too little or too much, fatigue or stress, impairs prefrontal function, whereas it is at its strongest when we are alert, safe, and interested. So let me show you some of what we've learned about how catecholamines do this at the cellular level. Most generally, I should say that at optimal levels of catecholamines, not too little, not too much, we have then strong prefrontal top-down regulation and mindful responses. But when we have too much, as occurs with stress, prefrontal goes rapidly offline. But at the same time, we are strengthening our primitive brain circuits, habitual responses by the basal ganglia, emotional responses by the amygdala, and also enhancing the sensory cortex. So this allows us to rapidly switch from a reflective to a reflexive state. And that may save your life when you're in danger, cut off on the highway, but it's really not helpful when you're taking a math exam or doing public speaking. And so what we've seen is that the prefrontal cortex has completely different needs than rest of brain, where I'll show you alpha 2A improves, alpha 1 impairs, and also beta 1 impairs. And it's the opposite for these other structures. And as norepinephrine has higher affinity for alpha 2 than alpha 1, the alpha 2 is what is predominating at low, safe, alert levels of norepinephrine, whereas this is what is at high levels during stress. So, first of all, catecholamines are absolutely essential for prefrontal functions, So if we deplete the prefrontal cortex of catecholamines, or if we block them, it's as if we've removed the cortex itself, removed the prefrontal cortex itself. So absolutely essential. And we now know this inverted U, how it comes about. And what we see with this increasing levels of norepinephrine and dopamine, with fatigue, we have too little. Optimally, I'll show you it's alpha 2A and optimal levels of D1. But then when we switch over into stress, now we start engaging alpha 1 receptors and too much D1, and now we're impairing prefrontal. So we can see this when we record from our delay cells, when we have too little norepinephrine alpha 2, the cells are unable to fire. When we have strong alpha 2 stimulation, we have really, this is the preferred direction, and this is one of the 7 non-preferred directions. So here's optimal firing. But now when we give high levels of norepinephrine through alpha 1, we suppress firing again. And so you really need to have it just right. We see the same thing with dopamine D1, but it's a touch more complex. Dopamine has fundamental excitatory actions that are needed to allow the neuron, the circuit, to fire at all. It then has sculpting actions where it gets rid of noise. But then with higher levels, such as during stress, now we again suppress firing. So let me show you what we've been learning about how this actually happens. Here is our synapse with NMDA glutamate receptors. And we've discovered something really fun, where NMDA receptors are normally clogged by magnesium. They have to sit in an electrified membrane to get rid of that magnesium and allow cells to talk to each other. What normally electrifies a membrane in a classic circuit is glutamate AMPA receptors. But in prefrontal, it's actually acetylcholine stimulating nicotinic receptors, nicotinic alpha 7 receptors specifically, which sit right in the synapse and electrify the membrane. And since acetylcholine is not released during deep sleep, these cells can't talk to each other and we are unconscious. But then we have acetylcholine release when we wake up, and now they can connect. I think that's really cool. Caffeine increases acetylcholine release. Obviously, cigarettes, this explains most of our behavior. But now let me tell you how we especially fall apart with stress, which is very much the other part of my behavior. So through NMDA receptors, we have not only sodium, but calcium. And we have this unique negative feedback in these newly evolved prefrontal cells, where, don't get scared by this, there is this genie here that's really evil. This is feed-forward calcium cyclic AMP signaling, where cyclic AMP drives more calcium release out of the smooth endoplasmic reticulum. Calcium drives more cyclic AMP production. And together they open these nearby potassium channels that I told you about before and disconnect the circuit. And this is really important for stopping seizures in a circuit where everybody's exciting each other. But you have to control it, and with fatigue, this is why you have weak function. With optimal levels of calcium, of arousal, we now have norepinephrine engaging alpha-2 and dopamine D1 to inhibit this process. So norepinephrine through alpha-2 inhibits cyclic AMP production, shuts off the genie, closes these channels, and allows them to connect. And dopamine D1 has this effect of phosphorylating the NMDA receptors, so they'll stay in the synapse and not hide below. And we can see that using something called immunoelectron microscopy. So here's the alpha-2 receptors, these little gold dots there. This is the synapse. And the red arrow points to this big gold particle is the HCN channel. So we can see this constellation right next to each other in the membrane near the synapse. D1 receptors are right in the synapse where they can have this effect. But they're also next to these HCN channels. And this is what is mediating their other actions. So dopamine D1 during stress drives this and opens these channels, and high levels of norepinephrine during stress engage alpha-1 and drive it through calcium. So now we have these opening and loss of firing and impaired function. And I'm blowing this up here to point out that protein kinase C is part of this calcium mechanism, and we'll see how relevant that is to lead poisoning and bipolar mania in just a moment. So that's part of these stress pathways. With chronic stress, we actually lose these connections, and we can prevent the loss of spines by either inhibiting pKa activity with guanfacine or pKc activity with something called chlorethrine. So let's see how this is relevant to diagnosis. First of all, just like in our animals, humans exposed to stress have impaired prefrontal function, either acutely, this is reduced dorsolateral activity, or with chronic stress exposure, the actual loss of gray matter in prefrontal cortex. And as I was forewarning you about, lead poisoning also causes loss of prefrontal gray matter, and lo and behold, lead mimics calcium. In fact, it's even more potent than calcium in activating protein kinase C. So we think this loss of gray matter may be due to this evil genie that lead drives even at very low levels. And then finally, bipolar mania. Husseini Manji and colleagues at the NIMH discovered that most treatments for bipolar disorder inhibit protein kinase C, including they did a proof of concept trial with high-dose tamoxifen, which at high doses inhibits PKC as well. And bipolar mania is explicitly associated with reduced activity of prefrontal in the right hemisphere. This is the work of Hilary Blumberg. So impaired insight, impaired regulation of emotion, and this is the exact same area that is slow to develop. Oh, really? This only says 15 minutes, 14 minutes, okay. So, because I didn't, well, anyway. So relevance to treatment, a whole variety of treatments optimize catecholamines in prefrontal. Guanfacine stimulating the alpha-2 receptor, and I showed you where they were, enhances prefrontal firing. And in monkeys, improves working memory, especially under distracting conditions, and improves impulse control. Jeff Newcorn's group has shown that guanfacine can activate, enhance activation of prefrontal in humans as well. And we see in animals that it can reverse the effects of either acute or chronic stress. And so approved for ADHD by the FDA back in 2009, but it's also uniquely helpful in kids who've been abused or traumatized, the work of Dan Connor. And I think it's because it helps in either direction, whereas stimulants can be harmful under those conditions. Atomoxetine blocks the norepinephrine transporter and increases both norepinephrine and dopamine in rodent prefrontal. We see a very narrow inverted U. This is 5, this is only 10. So I think that might be part of why this isn't that helpful, and that its enhancing effects involve both alpha-2 and D1 actions. Stimulants blocking both norepinephrine and dopamine transporters, and I wanted to quickly go over the work of Craig Berridge. We have been unable to ionophorese methylphenidate, but Craig Berridge has done wonderful work in rats showing that methylphenidate and amphetamines especially increase norepinephrine in prefrontal, even more than dopamine, and prefrontal more than in the nucleus accumbens. And the enhancing low doses improve working memory, whereas the higher doses do not. And these enhancing doses improve prefrontal firing in rats, whereas the higher doses don't. And the higher doses still are the ones that increase locomotion and actually take prefrontal offline. If he puts methylphenidate right into prefrontal, he sees the same inverted U, whereas in striatum, very little. And we can see an inverted U with methylphenidate in monkeys. These are older animals, and so less broad inverted U. But the beneficial effects are reversed by either an alpha-2 or a D1 antagonist. So both of these are playing a role. And I'd like to end with this one, showing that our work is suggesting new potential therapeutic mechanisms for strengthening these connections. Pfizer's just come up with a low affinity D1 agonist that might be helpful. I'm hoping that somebody can take advantage of alpha-7s. The nicotinic alpha-7 is not addictive, so hopefully that could be useful someday. And we're currently working on glutamate mGluR3, which has even better effects than guanfacine. So I'm hopeful that we might have new treatments in the future. And I'll end by thanking my many collaborators, Konstantinos Paspalis in particular for the EM, and Min Wang for the physiology. And thanks to all of you for your attention.
Thank you, Amy, for this wonderful talk. It's really the first time that I'm able to connect stress to lead and ADHD and other stuff, and that it all makes sense in the function of the prefrontal cortex. So wonderful talk, and hopeful that we might have new treatments in the future based on this work. That's really exciting. Who can I give the microphone for the first question? Tom.
Amy, very impressive coverage of very complicated material.
Thank you.
Thank you for that. My question is one of about individual variability within populations. You've given us a very nice description of how some of these various chemicals interact with one another and the impact on cell functioning, communication. And you've been very good, I think, in your work over the years in helping us to notice, this inverted U curve and that there's certain optimal points. But from the clinical point of view, one of the things that I think becomes particularly important is the question of how dosing optimal dosing varies from one individual to another. And I know a lot of the work you've done, of course, is with chimps. But I'm interested in how you address the variability, whether we're talking about different chimps in the group or different adults or children. How much variability do you see? Because I think that there is a tendency still for some people to want to prescribe medications for ADHD on a mg per kg basis. And the clinical experience for many of us is that that doesn't work very well. That there's some very small people who need bigger doses and some very big people who need very smaller doses. And I'm wondering, what advice can you give us about the degree of variability in terms of effective dosing for these various agents?
So we study rhesus monkeys, not chimps, where one isn't allowed to do research on chimps since they're apes. We also see that variability in monkeys. In fact, it's hard because with an inverted U, then you can't just combine everybody at one dose because they wash each other out. So we often have to really show our dose response curves. So many factors would influence that. Mm-hmm. Obviously, simple things like metabolism of the drug, the integrity of the blood-brain barrier, which is a big issue in situations where there's been inflammation, for example. How much of these catecholamines you'd actually be making on your own, how much receptor, what your endogenous state of your prefrontal cortex is, the environment, whether it's stressful, boring, etc. So many factors. So I think we're always stuck with the individual pharmacotherapy of what is really best for each individual. And sadly, that usually takes a while to figure out. And changes when the environment changes as well.
Thank you.
So hope it's evident to everyone why I thought we needed to have Amy Arnsten in our meeting, and I pursued getting her here for several years to make this happen. One of the things that happens in the clinic, particularly inner city clinics, when you talked about stress, and you talked more about acute stresses, but we see a lot of kids that are under chronic conditions of stress. They live continually under chronic conditions of stress. And they frequently have symptoms of ADHD. Virtually everybody in our clinic gets a diagnosis of ADHD. Mm-hmm. And people are never really sure whether it's ADHD or chronic stress. Sometimes people like to say, well, this isn't really ADHD. They're living under chronic stress, so it's not ADHD. And what to do from a therapeutic point of view as well. Obviously, reducing the chronic stress is the most important thing you can do. But if you're going to give drugs, what should you give? Should you give alpha-2 agonists, for example? And I wondered if you could just talk about that vis-a-vis the boundaries of the conditions, the developmental psychopathology, the developmental effects on the catecholamine system of living in chronic stress, and therapeutic implications of that.
Yeah, no, this is very much why we're doing this session today. And the idea that if a stimulant makes someone worse, that something like guanfacine might be the alternative that's needed as we balance these many things. And those kids might also have lead poisoning, the kinds of things that might have impacted. So when they're in utero, if their mom wasn't getting great health care, and so the prefrontal circuits weren't developing as optimally as they could. So many factors can contribute, and the hope is that we have treatments that give these kids a better shot.
And I think you said that stimulants only increase to the right, regardless of where you are on the curve, whereas guanfacine can adjust up or down. And I wondered if you could just augment about that? Because that's theoretically very important.
Yes, so I didn't have time to talk about guanfacine's other presynaptic actions, which are what most people know about. So they can actually reduce norepinephrine and dopamine release during stressful conditions, so normalizing that way. And postsynaptic alpha-2 receptors can impair the functioning of the amygdala, which in a stress situation can be helpful as well, normalizing amygdala function. So in a stress setting, there are multiple ways that an alpha-2 agonist could be useful for bringing that, here, I'll do it this way, the inverted U back to here from a stress state.
Can I interrupt, please? Thank you. I'm sorry, we have to stop now with the discussion, but keep your question for the break, probably. And I'm happy to introduce now and thank Amy for her wonderful talk. I introduce to you Joel Nigg. He's a professor of psychiatry and behavioral neuroscience at Oregon Health and Science University and director of the Division of Psychology and an author of a recent book on ADHD and the environment. And that's also the subject of his talk, combined with genes and biology and everything, in fact. It's a book about everything, I'm afraid. Joel, please.
Thank you, everybody. It's great to be here and great to be part of this symposium. Thanks for being here. The only conflict I have is the royalties from the aforementioned book, and there's more information about that online at my webpage as well. I want to give a little bit of background on why, first of all, why do we even talk about the environment in ADHD? Isn't it all genetic? And so I want to talk about that, and then give some examples of how we can evaluate the environment using genetics to figure out causal patterns in this very hard to untangle picture. And the first piece when we think about genes and environments with ADHD is thinking about the paradigm. What's our paradigm for psychopathology, or our even paradigm for thinking about how psychopathology develops? And a paradigm here doesn't mean a theory of everything, it just means that a good example. So the paradigm for how to tie your shoes is watching somebody tie them correctly, and that's how we teach kids to tie our shoes. We give them a good paradigm for how to do it, a good exemplar. And so the paradigm that's not correct for ADHD is to think of it as a paradigm like a metabolic disease. We find the gene, we solve the problem. This was the paradigm for too long for mental illness, and we now know that it's wrong. A second wrong paradigm is linear causality, that ADHD or psychopathology functions like a machine, that there's a cause and an effect in a linear fashion, kind of like in physics. Find the causal chain, interrupt it, and solve the disease. So the old view of ADHD and frankly of most psychopathology combined these into a single model that was linear, where there was a gene or maybe a couple of genes that did something to a brain circuit, and that led to the behavior problems that we call ADHD, and it was just a matter of finding the right circuit and the right gene. We now know it's much more complex. There's multiple genes, multiple circuits, and multiple environments, so we need a different paradigm. I'm going to try to talk today in the context of this type of a model, which is a bidirectional circular model where there's dynamic recursive effects, early environments. There's several examples of early environments up there, and they really could represent mother, father, or child exposures, pre and postnatal, that they have a pathway of effect, perhaps through inflammation or immunological response as one possible hypothesis, that there's epigenetic change happening, modulated by genetic. So the DNA is still there. It's still very, very important, and extremely important, as I'll emphasize, but it's not operating in isolation, but in conjunction with environmental inputs to modulate brain circuits and behavior in dynamic recursive fashion. Why do I think this is a better model? Why are we going to move beyond genetics to an integrated gene environment story perhaps in a better way than we did in the past with specific genes in specific environments rather than black boxes. There's several reasons. One is that a complex disease model is better than a simple disease model. I'll illustrate that in a second. That it's well known that the heritability term in twin studies includes gene-environment interactions. We don't know how much of that variance is due to those. It might be a little or a lot. And the third is the increasingly clear epigenetic insight that unlike the statistical picture, when you go to the biological picture, it's always gene by environment. It's never one or the other. You can parse them out statistically, but you can't parse them out that way biologically. Let me explain why the complex disease model is better. This is observable just even at the level of epidemiology. It's fairly apparent. Single gene disorders are deterministic. You have the gene for Huntington's disease or the wrong gene form, and you'll get the disease eventually. These types of diseases are rare, inevitably less than 1 in 10,000 in the population, often far rarer than that. There's a large increased risk in relatives, orders of magnitude increased risk in relatives. The example of Huntington's, the general population risk is less than 1 in 10,000, but if your sibling has Huntington's, your odds are 1 in 2. So that's a large increase in risk. Complex disease doesn't operate that way. It's probabilistic. The causal patterns are never fully deterministic. The diseases are common, always more than 1 in 500, almost always. A few things are on the border of that, but that's very typical. There's only a small increase in risk to relatives. So in the case of ADHD, the population risk is about 3 or 4%. If your sibling has ADHD, your risk is maybe 15% or something, or 20%. It's not hundreds of times greater. It's only double or triple as much. The model here is not Huntington's. The model is hypertension or other complex diseases, obesity. And so psychopathology is generally complex disease, not single gene disorder. But this may seem obvious, but it's extremely impactful when we think about how we think about and design our studies of environment. Let's talk about heritability a little more. The heritability of ADHD is high, about 70%. That's the variation between individuals. And this is just an example of twin data showing that identical twins have much higher concordance for ADHD than non-identical twins. And that's why from dozens of studies, we see that heritability calculates out in the 0.7 to 0.8 range. But heritability is not the same as inherited in this case when it's complex disease. And here's some data on tuberculosis and leprosy. And these, of course, are associated with infections. And I'm not claiming that ADHD is infectious, but that environments may modulate genetic liability. What we see here is that the identical twin and fraternal twin patterns for these infectious diseases look a lot like they do for ADHD and other psychopathologies. Why is that? Obviously, we're not inheriting these diseases. We're inheriting some sort of liability for them, some sort of propensity that then is activated or actualized in the environment. In the case of infectious disease, we now know what that is. That's the infective bacteria. But in the case of ADHD, we don't know what the environment is, but we can propose or argue that plausibly maybe ADHD and conditions like it are liability times environment phenomena rather than inherited as is or whole cloth phenomena. This makes more sense biologically. So that's kind of the framework or the logic here. And so we're arguing that ADHD be thought of as genetic and environmental liability interacting with the environment. This doesn't mean that we're going to have an environmental ADHD and a genetic ADHD. It doesn't mean, it means instead that we're going to have multiple routes. There's going to be very few cases where there's a gene of major effect or an environment of major effect. Obviously, there are some examples of extreme environments or extreme genetic effects, but they're not the main garden variety cases. And so this is the result that we have, this dynamic variable syndrome with multiple routes that we have to untangle and simplify. So this is a rationale for exploring the environment, again in ADHD, in conjunction with the heritable liability. If we accept this logic, which environments do we study and how do we decide if they're causal? There's lots of candidates. Many of you, probably like me, travel a lot, and when I used to be, when I was on the airplane, I don't make this mistake anymore, I would acknowledge that I did research on ADHD when I was talking to my neighbors on the plane. And inevitably, I found out in very strong terms what the cause was and what I should be studying. And the interesting thing is that many of the theories from my fellow airplane travelers were pretty similar to the theories that are either in the popular media or even in the scientific literatures. Let's run through them real quickly. Here's the first main theories I heard. And although they sound funny, there actually are literatures on each of these. There are literatures on secular variation in child problems related to economic problems and so on. We've seen some interesting studies on that. There certainly are very serious books written by knowledgeable people on the impact of pharmaceutical marketing. My apologies to the pharmaceutical representatives in the audience. And there certainly is some extensive writing, even if some of it is somewhat speculative, on the role of performance pressures on children starting school too young. We've seen the artifact of age in kindergarten kids as one driver of diagnoses that has to be taken into account. So there are actually literatures on these things and certainly strong opinions as well. So there's kind of a sociological dimension that we have to acknowledge. You've all heard of the caregiver problems, and certainly those who don't have ADHD kids are eager to conclude that it's the parenting of their neighbors that is the driver of it. Those of us who've tried to manage a kid with ADHD in the clinic or elsewhere know that they don't bring out the best in us, and that there are dynamics that can be negative, but that the original driver of this is probably child-driven due to the child's temperament or already ingrained difficulties. We actually don't know very much about very early life, one of the areas that we're eager to explore in our research. But by the time a child is 3 or 4 years of age, there's been very nice research done by Russ Barkley and others showing that this is often child-driven effects where the adult gets caught in a negative exchange that may perpetuate the problem but isn't the original initiation of the problem. And of course, there are well-established biological teratogenic events, extreme exposures. The Romanian orphan study is a great illustration of extreme trauma and a type of ADHD, and high levels of prenatal exposures to alcohol, lead, and so on. But these are increasingly relatively rare. These are not most of the cases of ADHD. Most of the cases of ADHD that we see don't have these obvious triggers. They've got something else more subtle. And so a lot of the focus now is turning to very common environments that may be harmful or at least risky to the population as a whole, but that only some individuals are susceptible to. A good example of this is violent screen media. We know that there's a causal relationship between violent media images and increased aggression in children, but only in some children. Only some children are susceptible to this effect. Not all children become more violent when they watch violent images on TV or video games, but some do. And why are they susceptible and not others? I'll show you in a few minutes some susceptibility data for lead and diet and ADHD. The important point about these types of environments is that they're very, very common. They're almost universal exposures in the population. So if there are gene-by-environment interactions here, they're going to look like heritability in a twin study. And that's there's a math behind that I have a spare slide for if you're curious about it during the discussion. But I'll just please accept that assertion for the moment that common environments are very, very important for common disease. And disease that's 3 or 4% in the population is common from the genetic point of view. And there certainly is a large literature correlating many of these environments with ADHD. There's large literature correlating ADHD with a range of toxin exposures, a range of dietary insults, and, of course, a range of perinatal events, some of them common, some of them relatively uncommon, some of them in the middle. There's been a lot of work recently on birth weight, for example, and increasingly on maternal metabolic state, maternal BMI, and a growing literature on paternal, even preconception exposure in parents affecting offspring that is of interest. So the environments, there's plenty of environmental correlative data here. In fact, these data are hard to ignore. There must be something to be said about them. We can't just brush them aside. How do we understand these findings? how do we determine if they're causal? There's a number of questions about the causality that I think have to be thought about. One is it plausible that low levels of unhealthy diet or non-poisonous levels of lead or and so on, that these can actually do harm? They don't seem very harmful to me. I feel like I can handle it. How can they hurt a little kid? And so the plausibility issue. The second is gene-environment correlations. We know now that many seemingly environmental effects actually are heritable. And so how do we sort out that genetic effect on the environment? And the third is it's almost impossible to ever measure all the confounders in a possible study. If a kid with ADHD has more lead in their blood, you can covary all kinds of things, but you never know for sure what caused what in a correlational human study. So those are real challenges. So how can we get around them? I'm going to use the example of lead. I was really happy when Amy and I corresponded prior to this symposium. I found that she was going to use lead as one of her examples for prefrontal effects because lead is a near and dear topic in our research and an exemplar environment that we've studied a lot to try to understand it as, again, not as necessarily even the most important environment, but as one exemplar of a well-understood environment with well-understood neurobiology and a long history of research that we can study. So I'm going to talk a little bit about this cohort here that has, what you can see here at the time we got the data, normal lead levels for our population in the United States. The normal lead level here, I've changed the scale from micrograms per deciliter to parts per billion to make it a little more relatable, but this would be 0.8 micrograms per deciliter or 8 parts per billion. And our ADHD kids on the far left have a little bit more, but not much more. And the actionable level from the CDC is 50 on this scale. So I'd have to do an Al Gore here. It's way up here, on the chart. It's a lot more. So these are supposedly low levels. But they're not low in evolutionary terms. We don't obviously know a lot about lead exposure in evolutionary times, but we know that lead wasn't mined out of the earth until about 6,000 years ago. And so we have some reason to believe that ancient humans 20 or 30,000 years ago probably didn't have as much lead in their environment as we do. Lead is everywhere in our environment now. The thousands of tons that have been mined are still out there, and about half of those tons are still out there that have ever been mined somewhere in the environment. And based on a little limited fossil data from ancient human fossils, there's some estimates that the prehistoric level of lead exposure was something around 0.2 parts per billion, which would make our current national average still an order of magnitude greater, even though we're very, very low compared to where we were 50 years ago. So the low levels are still plausible in some sense in evolutionary terms when we think about how did our brains evolve, for what environments did they expect when they were evolving. And many of our modern environments, from diet to media to lead, weren't in the evolutionary environment, so we have a kind of an open question about plausibility. So the bigger issue really is the causality, and there's been a number of interesting designs here. Surrogate pregnancy, a fascinating way to test environmental versus genetic effects. Anita Thaper's group has pioneered that work in ADHD and smoking. There's a range of sibling twin and adoption designs, some very, very nice work done by Ben Leahy and others using sibling designs where you can really, and we've done a little bit of this work too, you can really begin to separate out causal effects when two siblings come from the same mother, and you can isolate effects that are child-specific versus maternal and so on. There are natural stratification studies. The Dutch famine study is a classic study in the health world. The classic autism study that Mike Rutter was a collaborator on. I think Honda was the first author in a city in Japan that stopped vaccinations, and what did that do to autism rates? That's kind of a natural stratification experiment. The method that we're very fond of right now is called Mendelian randomization, where you utilize functional genotypes to let nature create the experiment for you. Let nature randomly modulate the biology so you can see if that has an effect in your causal theory. Now, of course, for some environments, we can do experiments. This is just an experiment showing that you can give fatty acids to mothers in a randomized experiment and see an effect. There's a number of those kind of studies. Fatty acid supplementation randomized trials show some benefit for ADHD. The effects are small, but they're there. These are causal effects because these are randomized trials. So we do know that there's causality for environments in the dietary world for ADHD because we can do randomized experiments. And the only argument we have is whether the experiment was done very well, whether the effect is big enough to be clinically relevant, and so on. But we can't argue about whether it's a causal test. And so that's very helpful in diet. But we obviously can't do randomized trials for lead or stress or trauma and so on. So this is helpful in the dietary world and therapeutic world, but not in much of the exposure world. But it's important to know that these dietary environments do have small, real but small causal effects. Dietary effects with their fatty acids, artificial food colors. We did a meta-analysis on this. These effect sizes here on the left, a medication effect size would be about a 1 on this scale. So you can see these effects are very small compared to medication effects. So dietary effects are there, but they're small. They're not going to be a cure-all for ADHD, but they might be part of the picture for some children. So kind of a modulated picture there is emerging, I think, in the literature, although still with some dispute about the size of the effect, but somewhere in that range of 0.1 to 0.3, which is a modest effect at the population level, but nonetheless worth noting for environments. The accumulation of different environments might be where we end up here. But how do we proceed on lead here? I want to highlight this because it's one of our exemplars for this symposium. And what we did is we replicated the association, we combined the samples, we designed a Mendelian randomization study. Dr. Arnsten mentioned calcium, but lead also has lots of interaction with iron, and iron is another important element in the diet, and there's a gene called the hemochromatosis gene that affects iron uptake and is also widely agreed to affect lead handling. We don't have an independent replication here. I want to acknowledge that before I go on. But we have well-characterized ADHD. We do have genotype frequencies that match the population, so there's not a special population here genetically. This is a typical community population, and the control group levels also match the population average. So we represent the population very well in our genotype frequencies, allele frequencies, and in our lead blood levels, which is encouraging. And we didn't have any extreme blood levels. All of the kids had blood levels that are safe, according to the CDC, but we're among the groups that have begun to show that there is no safe level of lead when it comes to ADHD, that almost any lead level is correlated with ADHD symptoms, and this is one of the studies that showed that. And we're able to control a gene-environment correlation statistically here, genotype environment correlation, not for all genes, of course, but at least for the gene that we studied. And we also, in this case, we controlled race ethnicity only by self-report. In other studies, we control it genetically. We can't control race genetically, but you can control ethnicity genetically. And here we only controlled it by self-reports. That's another limitation. But let me explain the logic of this design. The logic here is you have an experience. It could be alcohol exposure, it could be stress, it could be diet, it could be a toxicant, in this case lead. It has a biological process that it engages in that then leads to an outcome in our theory. Ordinarily, we try to study this by measuring covariates that might explain the effect. And in the case of lead, the literature, there are studies that have covariated. Almost anything you can imagine has been covariated. But you always have to worry about covariates you couldn't measure. So what you see in the slide here is that I've illustrated measured covariates, but also unmeasured covariates, ones you either might not know about or weren't able to measure. So the logic here is that if you have a functional gene variant that modulates that biological mediator and it's randomly distributed in the population, in other words, it's not correlated with your outcome and it's not correlated with your exposure, this creates a natural experiment. That functional gene alters the biological process. If that changes the correlation between experience and outcome, it's hard to explain that if it wasn't causal. And so a causal effect becomes the most parsimonious explanation. So in this case, we've hypothesized that one version of the gene would, and the story of oxidation as the mechanism here is based on one animal study, so that's a little speculative, but it was the logic that we used, that one of these gene types would lead to a larger effect on the biological process and a relatively large correlation of lead and ADHD, the other would not. And that's in fact what we saw, that there's a very strong correlation, a standardized beta 0.84 in one genotype, but less than half of that effect in the other genotype. This points to two major conclusions. One is that the lead effect is causal in humans. This was the first study to demonstrate this, even though there's many animal studies that show this type of effect. But the second is that some children are more susceptible than others. So this is also consistent with the susceptibility liability effect, a proof of concept, if you will, for that idea of ADHD. And thirdly, it raises the possibility that you could begin to screen for susceptibility. Now, that's going to introduce a whole bunch of ethical and policy issues that I won't get into, but in concept, you still could imagine this gene isn't ready for that. As I mentioned, we don't have independent replication and so on, but the proof of concept here that in theory one could begin to think about doing this. And in this case, there actually is animal research showing how the lead effect is modulated through epigenetic effects in the hippocampus, which is kind of a good, encouraging piece for the story. There's similar data using similar designs for food additives and for other pollutants. So this approach is beginning to be productive. In the last couple of minutes, I just want to show you a final idea here about how to separate or combine genetic and environment effects, and that's through the use of a direct measure, molecular measure of genetic liability. And we do this with what's called a polygenic score. And the logic here is on the left that we have a discovery sample. In this case, we use large discovery samples, tens of thousands of individuals from the Psychiatric Genetics Consortium, ADHD case control, depression case control, bipolar case control. So we can create odds ratios for all the SNPs in the genome in terms of their association with the disorder from these very large samples. We take those odds ratios to our local sample here, and we can then create, through an equation, we can calculate just by essentially adding up all the alleles in their odds ratio, multiplied by their odds ratios. Every child gets a score of their genetic liability for ADHD, bipolar, depression, or other disorders. And there's a large number of these that can now be calculated. And we can then take that score. It's a continuous quantifiable measure of genetic liability. Now, we have to remember that these common gene variants are not all of the genetic effects of ADHD, but they're one of the domains of genetic effect in ADHD that we are now controlling. And then we can look at that in relation to various elements of the ADHD phenotype. It's a complex phenotype, and Dr. Arnsten mentioned arousal, and we've got a very extended measure of arousal here using some computational methods and computerized tasks to get at our version of arousal. This is not necessarily stress arousal, but it's a measure of arousal. And we also have measures of working memory and measures of irritability or anger susceptibility in children with ADHD. And we've shown that these create subgroups of kids that are reliably identified, clinical subgroups. I've got a whole other talk I give on emotion dysregulation as a subgroup of ADHD. And that the ADHD genetic effect here is independent of the mood effect, so these elements are part of the ADHD syndrome. But then importantly, when we combine, in this case, maternal prenatal stress, maternal smoking and drug use, and perinatal problems, we can see that the genetic liability, in this case, my hypothesis actually didn't work because there was no interaction, but we did see additive effects here of the environmental effect, prenatal stressors, and ADHD genetic risk, both on ADHD and on the emotional dysregulation of ADHD, leading to some hypotheses that Amy Arnsten will tell me what this means for the brain and the neurotransmitters that may be relevant to future treatments and to stress effects and some of what she talked about. So this is the work that we're very interested in pursuing and developing, both in relation to lead and stress and early effects. There's a number of caveats to all of this. We have a long way to go to specify the genetic liability and the exposures. The heterogeneity within ADHD is extremely important. We'll hear a little bit about that in the final talk. And the overlap of different disorders is extremely important as well. None of this is necessarily specific to ADHD, but I want to conclude by pointing out that there is evidence now for a causal role of some environments for ADHD. Our big task now is to separate which environments are not causal, some are not, some are artifacts of genetic effects, and which ones are, and then how big are those effects. So the effect sizes still remain to be determined. And so I really recommend when it comes to ADHD in the environment, a real balance of openness, it's not all genetic, and caution. There obviously are some exaggerated claims and enthusiasms, but there really are some environmental causal effects, and this opens hope for eventually identifying reversible causal inputs. We may be able to help ADHD faster by reversing environments that are specific and identifiable. Jeff Newcorn already mentioned chronic stressors that are hard to change, but there may be others that are easier to change and target, or there may be simple reversal cures for some of these that may come faster than modifying the genome. I want to thank my collaborators as well and all of you for being here. Thank you. I just want to say that thing turned to 000 right as I finished. That was really cool.
Timing is not your problem. Thank you very much, Joe. It's exciting to hear more about LEAD and the possible role or the established role in ADHD and maybe other disorders. So my question would be, I learned at school that LEAD is in the tubes of the water supply. And since we knew this from the 60s, they've been trying to change that in my country. Is this true in the United States?
Yes.
So since when, and how come that still the LEAD is measurable in the blood of our children?
We have been able to find measurable lead levels in 99% of the children we test, and that's consistent with what the CDC says. United States. The United States is probably not as far along as the Netherlands, but we're still one of the best in the world as far as lead exposure compared to other countries. We still have lead exposure from old water infrastructure, as you mentioned, but also from old housing stock. Lead stays in the environment a long time in the dirt and schoolyards. It's even in food because of air pollution from smokestacks, airplane exhaust. There's a range of exposure routes still. But the permanence of it in the environment is one of the challenges. Lead that was released decades ago is still there. And so one recommendation that's been made is to replace all the housing stock built before 1940. That would cost about $60 or $70 billion. Those calculations have been done. There are people arguing that's one of the best public health investments we could make. So there's a range of, but of course replacing water infrastructure is a big one. We've had crises in the United States with water exposures in Flint, actually in Portland. In the last few years, this has been a big topic in the media. But those are high exposures, and I'm also talking about the average exposures even without those disastrous exposures. So this is an ongoing issue. We're way better than we were 30 years ago due to paint and auto exhaust improvements, but we still have to fix water and housing. Yeah.
Joel, thank you for an outstanding talk, emphasizing once again the role of modifiable epigenetic risk factors, where we can make a difference on these epigenetic mechanisms that can potentially either be damaging, but can also be strengthening for the children we take care of. So the things we can do to tell parents about decreasing maternal weight, watching your maternal diet, decreasing nicotine exposure, the influence of positive life events on positive epigenetic mechanisms, and the damaging effects of stressful life events on damaging epigenetic mechanisms. Something we don't tend to talk about when we're teaching, when we're talking to our patients, and so I think it's just incredibly powerful to hear you stand up and talk about it. So thank you.
Thanks for that comment. That's my whole book. It's what are the positive things you can do, and there's a list of them. So it's very important that we can take a positive approach. It's not all risk. It also is protective.
Yeah, I want to thank you also for great talk, and your work has obviously been very influential over the years. Two public policy questions come up. One has to do with the legalization of marijuana, and you in Oregon are probably the first in the forefront among other states. Mm-hmm. And if anything, do we have any direction to go in thinking about genetic risk, about not only exposure to marijuana for purposes of psychosis, but also for maybe explaining adult onset or later onset of ADHD and related symptoms? The second is the one that I think has been under-discussed is, and again, not to point fingers, but to say, and align with what Greg just said, is maternal alcohol and the role of alcohol and alcohol exposure, not only in this country, but I guess looking at cross-national data and thinking about, especially in the early days of pregnancy, before you know. And I wondered if you could comment on those two toxicants, if you will.
Yeah, certainly marijuana, you're right. Oregon is one of the first states to legalize. We now have legalized recreational pot, and I have a couple of colleagues at OHSU that are experts on this area and adolescent brain development and marijuana, and what I've learned from them is what we don't know about the effect of marijuana on the adolescent brain would fill volumes. And so this is an area of research that needs to be emphasized and highlighted, and I think we had a New York Times article on this just a couple days ago relating to psychosis that you may be thinking of in your question, but there's also this question about other effects of marijuana use in the adolescent brain, and I think it's a hugely important unknown that needs to be researched. The issue of prenatal health and maternal health is probably still, this was true 30 years ago, it's probably even more true now, that one of the best public health investments we can make is to improve prenatal health for women, and that includes stress, it includes drug and alcohol and cigarette exposure. How can we ameliorate those effects? There's increasing evidence that vitamin C can dampen some of the smoking effects. There's evidence that omega-3s can dampen some of the metabolic syndrome effects in women. I don't have any way to ameliorate alcohol effects, and I don't know a lot about low-level first month of pregnancy effects that you allude to for alcohol. As you say, most women now are able to stop alcohol once they know they're pregnant. And then these toxin exposures that are kind of a nightmare because no one knows how to prevent them. And can we ameliorate those? But we do know that there's interactions among all these risk factors. So we know that stress interacts with toxin exposure, for example, or that inflammatory effects are common to many of these risks. And so the fact that they interact, I think, gives a great deal of hope to parents and clinicians that you may not be able to solve all of these risk factors, but if you solve even some of them, you may change the dynamic because they're multiplicative most likely. Yeah, thanks for that point.
Okay. All right. Thank you very much, Joe. Warm applause for Joe. And then I'm happy to introduce you to the third speaker. It's Kathleen Merikangas. She is a senior investigator at the National Institute of Mental Health and in the intramural research program specifically. And she will speak about the comorbidity of attention deficit disorder and physical conditions, explanations, and impact. Floor is yours.
There's a timer right there, right? Okay. Is that the, no, timer.
No, I'll be your clock. Let's see.
You'll be my clock, okay?
Yeah, I'll sign to you when it's done.
Yes.
So, here you go.
Thank you. Okay. Morning, and thank you for those wonderful talks that preceded me on the biology and environmental factors. I go to a lot of different meetings because I'm trained in epidemiology and genetics, and we're supposed to know about everything, the epidemiology of all the disorders we study. So it becomes very complicated when we go to meetings in specialty groups, because this is one of the first meetings I've gone to where we're actually paying a lot of attention to environmental risk factors for the onset of attention deficit disorder rather than focusing exclusively on the genetic underpinnings of some of the conditions. So I think there's a lot of hope in being able to treat and prevent attention deficit disorder and related conditions. So I'm going to be talking about comorbidity of physical conditions with ADHD, and I'll be talking about the different causes of these patterns of comorbidity and what some of the implications are, not only for etiology, but also for treatment and for public health. So my comments are going to be much broader. I'm not going to be presenting a lot of our own research on this topic, which we're doing now at the intramural program of the National Institute of Mental Health. I would like to give a shout out to Dr. Judy Rappaport, who set up these programs many, many years ago, and Dr. Philip Shaw, who will be here speaking tomorrow, because they've set up a wonderful environment for us to bring people into the clinical center of the NIMH and focus broadly on causes and risk factors for these conditions. So first is the way of background. Comorbidity is a term that we use every day. Now, every journal we pick up has at least 3 or 4 articles that at least mention comorbidity. But when we started in the field, I'm trained primarily as an epidemiologist. I was a postdoc of Myrna Weissman at Yale. Dr. Biederman will remember this because when we went and did population-based studies, started to look at the epidemiologic catchment area data, we found that people met criteria for multiple disorders. And then the question was, what do you do? Do you count all these people 4 and 5 times when you're estimating base rates of conditions? And just at the time we were struggling with this question, we became aware of the work by Alvin Feinstein, who led the Robert Wood Johnson Clinical Scholars Program. I don't know if Andy Nierenberg's here this morning, but he was a colleague at NIMH who was one of the many scholars across the country who was trained to develop strong clinical research as part of his medical training. So at that time, Dr. Feinstein had written an article, and this was in 1970, talking about comorbidity, and defined comorbidity as a distinct clinical entity that's existed or may co-occur during the clinical course of a patient who has an index disease. So when we spoke with him about this and asked him whether this really applied to the problem that we were having with people meeting multiple conditions, he said, no, you're trying to think of how are they involved, how are these 2 conditions involved etiologically. But what he essentially was advocating people to do was to characterize people according to all of the conditions that they may manifest. And particularly he was interested in the impact of this on clinical trials. Because if you have a person who has side effect A or B, or another condition, then the results of your clinical trial will be spurious. So he said, you don't have to think about causality. You need to characterize people comprehensively. And this is really the background for what I'm going to be talking about today, as you'll hear in a few moments. So now let's fast forward to this wonderful review of medical and mental comorbidity that was sponsored by the Robert Wood Johnson Foundation. And one of my colleagues, Dr. Ben Druss and Elaine Walker at Emory, were tasked with doing a comprehensive review of medical mental comorbidity, particularly as it impacts on treatment and services. So they did a beautiful review of this. It's about 10 years old, and started to look at differential patterns of comorbidity and what are the implications when we treat people in the general population. They focused primarily on very severe mental disorders in adults, or what's known as SMI, by the terms of when SAMHSA is trying to enumerate people. But this review concluded that the mismatch between clinical reality of medical mental comorbidity and a medical care system that separates these domains is a major future challenge to medical care in America. Now, I learned this when I was back at Yale, and the head of the Substance Abuse Administration asked us to do a population-based study of people who were homeless. Now, that's been accomplished subsequently, but that's a very complicated question, because how do you enumerate all the homeless people in a particular area? How do you do a comprehensive evaluation? But what I learned from undertaking this project was that the Substance Abuse Administration was under completely separate funding and administration. So what they wanted to do was to say these homeless people are either mentally ill or they have substance use disorders, because then the funding would be in one administrative pocket rather than the other. And this is when I became so aware of the SAMHSA definitions that they're forced to characterize serious mental disorders and serious emotional disorders, that you exclude people with substance abuse, you exclude children with autism from serious emotional disorders. And this is quite relevant to care, as I'll say later on. But this idea that we separate all these domains in the healthcare system is really important because it does affect our ability to do the kind of research that we're talking about at this meeting and the ongoing studies that we're doing in treatment and etiology. So one of the things we've learned from our large-scale population-based studies is comorbidity is probably the major marker of severity of conditions. People with comorbid physical disorders have increased mortality when we follow them over time, greater health care costs, poorer treatment response, and greater functional impairment. So comorbidity is a marker of severity. Now, moreover, when we're trying to study comorbidity, and I've done a comprehensive review of this question, it is really, really challenging. Because when we put together data, aggregate data across the world, we find such variability, as any other mega or meta-analysis that people have done. We see that there's huge sampling variability. I don't know if this is working at all, I don't think so. That a lot of the data that we have is based on either parent or child reports that their child has one of these conditions. The diagnoses in the registries that we're now working with the Kaiser Registry on adult ADHD. We're working with registries at University of Pennsylvania and then some international studies. We see that we rely strictly on ICD or DSM codes, and that's the only information we have when we're trying to look at patterns of comorbidity. And of course, as we know, and we've heard throughout this meeting, there's a real spectrum underlying these conditions. So this is a bit misleading to just look at comorbidity or prevalence rates strictly by the DSM codes from the different registries that we work with. Certainly the biggest challenge has been non-comparable information on mental and physical conditions, and this is a challenge for those of us who are epidemiologists because we can't possibly go out into the population and collect the full spectrum of conditions that children suffer from in terms of cardiovascular disease, neurologic conditions, headache conditions, and then moving beneath that to the components of these conditions such as sleep, patterns, and disorders, and so forth. So it's just beyond the scope of any study to comprehensively characterize both mental and physical conditions. So that's a real challenge, and the question is, what are some of the solutions to our inability to characterize people comprehensively with respect to medical and mental conditions? The other limitations that we've seen again and again, there's a limited range of mental and physical conditions, and some of the data I'll show you today, some people will focus on some medical conditions and not others. They'll use different terms and different diagnostic criteria. So it's really difficult to put this evidence together. Most of the data is cross-sectional. So if we really want to learn about the kind of things we've heard earlier today, for instance, the work on lead over time, we really need prospective data. Most of the data we work with are cross-sectional, and we're forced to rely on retrospective recall of the onset of these conditions. And as I'll talk about in a few minutes, most of the conditions we see that may underlie attention deficit disorder may actually emerge or be influential prenatally or early in development. So it's very difficult to do retrospective assessment of these issues. And then finally, small numbers of cases. So if we really want to delve down and move beyond neurologic disorders, psychiatric disorders, cardiovascular disorders, we look at these specific conditions, the rates are very rare of a lot of these conditions. So then even if we look at neurologic disorders and seizures, we've seen that some seizure syndromes are related to ADHD and others are not, but the rates of these are so low that you really can't accumulate sufficient number of cases in general population studies. All this is to say, as an epidemiologist, it's very complicated data out there, but I think being aware of these kinds of methodologic limitations is very important. Now, I'd like to now turn to the evidence that we've accumulated over the years. I'll talk about a couple of studies. Fortunately, we published some of this work, but we're now trying to replicate it in new studies that are just emerging across the world. There's a number of studies where data are just being released, where we're working in a consortia of child mental health epidemiology to build the kinds of models that we'd like to build on these patterns in the general population. Now, this is a very important slide for those of us who are epidemiologists. It's Epidemiology 101. It's what we call the tip of the iceberg phenomenon. I actually found a pretty nice picture of an iceberg here. But what we see here, there's 2 points I wanted to make. And the first point is written at the top of the slide. I primarily study bipolar disorder, and at the NIMH, we've now spent over a decade collecting families of people who have bipolar spectrum disorder. And of course, we've gotten into looking at attention deficit disorder kind of through looking at families with bipolar disorder and population samples. But what I've seen at the NIMH is the way we sample people, we really don't have very much that's generalizable, because we advertise. Since 9/11, we now have a big wall around the NIH campus, where any of you who know NIH, most of you do, in the intramural program, it used to be the people from Bethesda and the region could just walk across the campus to get in the metro. But now we have a wall. My friends joke that they have to practically have a colonoscopy to get on campus, because it requires, you have to have, they're not doing fingerprinting yet, but close to that. So it's very difficult, and I think this has really been a challenge for those of us who want to study local community samples. So I've spent the last decade trying to collect data on families from the broader Washington area who are identified not because they have a condition, but because they're interested in looking at health and behavior. So we've now collected families, more than 600 families that have been comprehensively characterized for a broad range of phenotypes. But what we found is when I started this study, and that was at the time I came to NIMH intramural with Dennis Charney from Yale, where we started a mood disorder program. You mentioned Husseini Manji earlier. Carlos Zarate came at the time, and I've been really working very closely with Carlos and other people. But what we've seen in the last decade is there's been a huge, growing divergence between what we see in the population and what we see in the clinics. And when we look across the US, we see that about 60 to 70% of people with severe bipolar disorder or severe depression never see a mental health specialist. And as you know, you're seeing more and more, and this has been an issue for attention deficit disorder for a long time. It's screened in pediatric settings, internal medicine, but we're having the same thing with depression now. They're doing a screening scale in all pediatric settings, and then the question is, what about comorbidity? How do we put it together? You can't have the same expertise in general primary care that you're going to have in mental health settings. So this has led to growing divergence between the kind of samples we study in our samples. And so by going to the population, we found that about 60% of the people in our study are being treated with lithium or another of these agents, mood stabilizers, by people who are not mental health experts. So I think that's a really big issue, because first, nobody can really have the wherewithal and the clinical expertise to do these comprehensive evaluations. But with the lack of clinicians, even in the Boston area, that was a very famous article in the Boston Globe talking about taking more than a year to find an expert to treat a child newly diagnosed with ADHD. So this is a big challenge we have. But for us as epidemiologists, it's even a greater challenge, because we worry about the representativeness of samples. And if all of our samples are coming from clinical sites and we don't know how atypical they are, then that may lead to spurious conclusions on associations and so forth. And indeed, one of the most important factors that gets you into mental health treatment is comorbidity. So samples of youth being treated with ADHD, adults certainly with ADHD, they are more likely to have multiple comorbid disorders than those from the general population. So the second point I wanted to make is this subclinical phenomenology. When I was a postdoc at Yale, a professor named Jules Angst from Switzerland came and gave a talk which I was so impressed with his way of thinking. He's now 92 years old. He's in the Burgholzli in Switzerland, and he was the first to delimit bipolar disorder from unipolar disorder based on familial aggregation of mania and depression. And they separated out unipolar depression from bipolar depression, from manic episodes. And I started to work with him because he always said these DSM criteria you have, they don't make sense. There's overlap between schizophrenia and bipolar disorder and so forth and so on, and you really need to look at spectra. So at that time, he did a very large, well, it's not large numbers, but for Switzerland, it was a big step to go into the general population and follow people. Now we've followed them for 40 years. So this sample has been followed phenomenologically. And it was his model that led us to capture phenomenology in most of the studies we do. So many of the studies I'll be talking about today, we don't just rely on SCID-based DSM. We've developed tools to look at the full spectrum of these conditions. And now those tools now are available to people to look at the spectra of expression in general population samples of people who do not come for treatment. Okay, now the first study I'll talk about is the National Comorbidity Survey, Adolescent Supplement, on which I was the principal investigator along with Ron Kessler from Harvard. And what we did was extend the National Comorbidity Survey replication in adults to youth. And I worked with Dan Offord, very well-known, he's subsequently deceased, social psychiatrist who started the Offord Center at McMaster in Canada. And now I'm working with one of his young trainees, Kathy Georgiadis, who is now the PI on the Ontario Child Health Survey, and we're now following his tradition of social epidemiology. And he always admonished me for looking at all these DSM disorders. He said, you need to look at impairment because that's the important thing in kids. If we want to follow kids, look at impairment. Diagnoses are important, but the main outcome that we want to follow up on is impairment. So the Ontario Child Health Survey places a lot of effort on looking on functional implications of these kinds of symptoms and syndromes. So what we did in the NCSA to look at physical and mental comorbidity was we captured a lot of data on chronic conditions. So we used a questionnaire, and again, it's all questionnaire-based, so of course that can be unreliable. So if a parent tells, I study migraine, that's one of the major areas of my research, and a lot of people will say they have migraine, but they don't meet criteria. Migraine's used synonymously with severe headaches. So this is true for many of the conditions, the medical conditions. It's may not be accurately depicted that would be concordant with what a clinician would diagnose in terms of the medical disorders. But the first point is, and then I won't talk about this again, Dr. Nigg asked me, are you going to talk about psychiatric comorbidity? And yes, this is the work we've been doing, not only in NCSA, but a lot of studies across the years. And what this shows is among those adolescents, ages 13 to 18, and that's important, it's adolescent sample, we see that 80% of adolescents in the general population who meet full criteria for attention deficit disorder have at least one comorbid mental disorder. And then if you look at the different conditions, we characterize these by class. So almost half will suffer from major depression. 60% will have behavior disorders or substance abuse. And Ron Kessler and Jen Green wrote a very nice paper on these data, looking at the order of onset of these conditions, and indeed for attention deficit disorder, usually the ADHD is temporally primary. Now that's because, partly it's artifactual because you have to have the age of onset has to occur before, well, it used to be 5, 6, and now there's been a shift to age 12. But because it's required to be a childhood condition, then of course it's going to be temporarily primary. So we have to think about that, with some skepticism, because onset of ADHD is insidious, as we know, and it may occur later in life as we're beginning to study this in adult populations as well. So let me just, in the next few minutes, summarize some of the findings from the different studies, but broadly to give you an idea of what the challenges are, but nevertheless we can conclude, first, that there is strong comorbidity between physical conditions and mental disorders, particularly with ADHD. Now, ADHD is a kind of special case because it has to do with brain functions, as we nicely heard. So a lot of what they call comorbid conditions here are disorders of the brain that may be truly the same underlying diathesis as attention deficit symptoms in these people. So it's kind of artifactual to say that the neurologic or neurodevelopmental disorders are comorbid because it may be part of the same underlying set of risk factors, and I'll talk about that in a moment. But what we found in the NCSA was that all of the conditions that we see here, allergies, headache, asthma, obesity, et cetera, were more common in youth with ADHD compared to those without. But then when we controlled for other physical conditions and comorbid medical conditions, the only things that really jumped out were enuresis and stomach and bowel conditions. And then when we controlled for depression, that even went away. So the only thing that was unique to ADHD in that sample was enuresis, which is very interesting disorder. Disinhibition occurs, developmental condition, but that's been widely shown in other studies, both clinical and community samples. But there hasn't been a whole lot of work paying attention to that. So if that's the unique physical trait or characteristic that we see with ADHD, I think that might be important to characterize people by that, to see how's that related to brain function, how's it related to cognition, how's it related to learning, and so forth and so on. And then the second point I wanted to make about the NCSA here is that we see that there's been specificity for the physical conditions associated with ADHD. So mainly we see learning disabilities, enuresis, and developmental conditions, but if we look at other disorders, we see that learning disabilities are related to all of the conditions we study except anxiety. So we also see learning disability associated with behavior disorder and mood disorder. So we do see, again, the developmental and disorders and enuresis are specifically associated with ADHD. Now, moving forward to look at just these bivariate associations, we then looked at the number of physical conditions that youth have. So just like mental disorders, where the average child with ADHD in the population, not necessarily being treated, has 3 or more disorders, we see here that about 60% of kids with ADHD have 2 or more medical conditions. So this doesn't even put into the picture comorbid depression, anxiety, behavior disorders, and so forth. So what we're really dealing with is kids who have multimorbidity in the general population, not necessarily an artifact of treatment. So then we started to work with the Philadelphia Neurodevelopmental Cohort Study. I was involved with this study from the start, where we used the tools from our intramural program, the screening tools for mental disorders and physical disorders. In this study, the data are now publicly available, and we published a paper on physical mental comorbidity in this sample. It's a sample of 9,000 youth who were identified at the Children's Hospital of Philadelphia, many for primary care, but others for specialty care. So what we wanted to do was to see if the findings from population-based samples would also hold up in samples where the youth had been diagnosed by a pediatrician or some expert, and indeed we found the same thing. We found that the developmental and neurologic conditions were associated with ADHD. Again, developmental conditions are related to many other kinds of disorders over the lifespan, but particularly the neurologic and CNS disorders. And then we did see some specificity as well. So the second point I wanted to make here was the more severe the disorder, the more likely it was to be associated with these different disorders. So if you look at this group in the middle, youth with ADHD were not only more likely to have a medical condition, but also a more severe medical condition. Okay, so these are the basic findings from the epidemiologic studies, the population studies, and we're now doing more work, I'll show you in a moment, with the Child Mind Institute study called the Healthy Brain Initiative, where our goal is to collect 10,000 youth. So we started to look at some of our preliminary data there to look at subtypes. And this is a sample. It's Michael Millam is the PI of the study. There's a very wonderful multidisciplinary study team involved in this study. So we wanted to look at the elimination disorders. I was interested in following up to see if they found the same thing, and indeed, they found differences by subtypes of ADHD. So here we show, and these are just very preliminary, we showed that hyperactivity is related to the elimination disorders, but not the inattentive and combined subtypes. Those subtypes, the inattentive tends to be more related to communication deficits. So there was some heterogeneity by the subtypes of ADHD in these studies, which I think is very important to look at the heterogeneity of ADHD in these studies as well. Okay, now let me move on to the explanations for comorbidity, and you set up the stage very nicely, Dr. Nigg, because he talked about causality of lead and so forth. This is a slide that I've had probably since 1984, 5, talking about the two broad sets of explanations for comorbidity, and you've already nicely talked about that. So are there common risk factors that lead to disparate manifestations? Enuresis, is it different in kids with ADHD and not? But the two different explanations could be a causal mechanism, or it could be common etiology. Does having ADHD lead to depression? That's a widely studied question. Does it lead to depression? Does it lead to substance abuse? And that's the kind of causal mechanism. I have to note that JAMA journals no longer allow us to use the word causal. So I've reviewed some Mendelian randomization studies, and they say causal, and they eliminated it. So we have to be able to deal with that. So it's in quotes. We don't really believe it's cause. We hope we can get to that, but that's not, we're not really overstating that case. So these are some mechanisms, and we nicely heard some of them. Causal could be that the learning disability inhibits development of attention or motor control. Seizures or epilepsy lead to executive motor function impairment, or common risk factors such as the lead, pre-perinatal factors, a lot of maternal behaviors, early environmental exposures, common genetic factors. Again, this is what you just talked about, Dr. Nigg, and then risky health behaviors. So these are the possible mechanisms that we can study. So what are some of the implications of this pervasive physical comorbidity and attention deficit disorders in the population? Well, I want to go back to Dr. Feinstein. He said neglective comorbidity can lead to inaccurate estimates of morbidity and mortality. It can lead to misleading fatality rates when you're studying, for instance, stroke, if you don't characterize the comorbid conditions, and spurious results of clinical trials. And I would add to this, spurious results of any research we do if we don't pay attention to this. We put a lot of effort into neuroimaging. It's very costly, and it's time-consuming. It's a lot of effort for the child and for the researchers, but most of the studies do not look at patterns of comorbidity, or they exclude kids who have depression or learning disabilities, and then have typically developing kids, whatever that is. So I think what I'm going to say in a few minutes, we really need to look at specificity. Okay, all right, so I just have a couple more slides to just talk about the implications. So this slide is from a paper that an editorial that we published in JAMA Psychiatry. And after I led a work group on serious emotional disorders for the Institute of Medicine that was funded by SAMHSA, to say, what should we do in the next decades if we're going to be going to the population? And after a lot of thought and input from many, many experts in the world, we came up with this notion that we should be counting children, people, not disorders. We've kind of been there, done that. We know the rates of ADHD, and they can be all over the place depending on how we do the study. But we really need to look at the number of children who are impaired, and then start with that and say, what is leading to their impairment? And this is kind of a, I think it's a challenge to those of us in epidemiology, but this is probably the only way to deal with this. So secondly, we need to look at comorbidity either as a confounder in our studies. It should be considered in our studies where we put a lot of time and effort to careful clinical trials, neuroimaging, genetics, and so forth. We really need to look at the overlap between these conditions. So both to understand patterns of comorbidity, but also as a confounder in our studies. And more broadly, we need to shift from the diagnostic single care approach to collaborative care models, which I'm happy to say the Affordable Health Care Act has one of the most beautiful descriptions of how we should be caring for people in the population, particularly children who suffer from ADHD, but also may have seizures, may have depression, maybe have substance abuse. They are now starting centers across the country where collaborative care is the standard. So you don't treat the child just for coming for ADHD or coming for substance abuse or coming for this or that. They are all treated for all of the conditions that they come for comprehensively in the same centers. And Kooij is funding a lot of this work. So this is I'm not going to show you this language, but the language is actually beautifully characterized by the Affordable Care Act. And now, ironically, the children who are on Medicaid are being evaluated by whether they have comprehensive care settings. I just gave grand rounds in Texas, and all the universities there are now banding together to do work, comprehensive care of youth and adults. Okay, finally, so what do we do with comorbidity when we have professional societies? This has been my challenge for the last decade. This is a meeting on attention deficit disorder. I study bipolar disorder, so I go to ISBD, which is in Australia. I go to ISAD, which is the International Society for Affective Disorders. I go to genetics meetings because I'm trained in genetics. I go to epidemiology meetings. I go to ACNP. I go to all these meetings, and this shows all the organizations we have. And I think it's great because we're learning more and more about ADHD. So as we learn more, we have to delve even deeper. So we can't go to broad meetings where we can really get the specialty and delve deeply into causal mechanisms. But at the same time, this has really challenged our ability for advocacy. Because we have each of these organizations, and as you might imagine, ADAA, the Anxiety Disorder Association of America, doesn't have as much advocacy of people with other conditions where they're more likely to demand care for their families and for their children. So there's somehow we need to balance. I don't have an answer, but we need to balance the increasing specialization of our fields with the broader issues of impairment, broader issues of the specialty research and care in the population. So let me just end with a couple quotes here by people from long ago who advocated for the same thing I'm advocating here. Leon Eisenberg said, we should be treating children in the medical settings and psychiatric settings together, and we should also be studying neural mechanisms by looking at the context, the physiologic context of brain and body. Thank you. Oh, yeah. Yeah, that's good.
Thank you very much on this important subject of the mental and the physical connection that we have neglected for way too long, I think, in medicine. There's time for maybe one or two questions. Yeah, please.
Thank you. I've long thought that we often think in groups more than individually, even when you come to societal problems and thinking about this poor person would respond to this method, this poor person would respond to another method to help them out as a society. So I take that message from this and the way to think about it. But the other thing is when we talk about outcomes, you're talking about impairment. I know that has measurable abilities to think about impairment, but we don't often put the phenomenon of suffering in the mix either. And I think it ought to be integrated into the thinking.
No, absolutely. And I study mood disorders and, of course, and anxiety disorders. And subjective distress is as important as impairment. Those are the two criteria. So thanks for reminding me of that.
Can I ask a question that pertains to more than Dr. Merikangas' presentation? We into that part of the discussion for the entire morning? Okay, well, maybe Kathleen can. First off, I'd like to say, I normally don't say that that was a wonderful symposium because they're all great, but that really was a very wonderful symposium and complete embarrassment of riches. So thank you, all three of you. That was fabulous. My question is a bit of, it comes mostly from Joel's work. It's a bit of a paradox, which is, and it's illustrated nicely in the problem of, well, in the use of polygenic risk scores to try and identify genetic risks that might interact with environmental risks. Why is there a paradox? Because polygenic risk scores tend to reflect those most strongly associated genes, and you would expect that the exact opposite, that genes that are interacting with environmental risk factors would be variably expressed in relationship to the phenotype, only in the presence of the environment. So the question that I would have is, how do we get the kind of genomic, like we'll probably need genome-wide approaches, and that will be a challenge because we'll need genome-wide environmental exposures along with our genome-wide genetic data. Now, Kathleen's looking at me like
I could start with that. I think it's a broader discussion. Well, polygene risk scores, when we look at them, I believe, I don't know if Steve Ferrone's here because he's on the PGC group for ADHD, but I believe there's 12 loci for ADHD at this point. So of all the loci, we have 12 that supersede the significance levels. And then one of the assumptions of Mendelian randomization is your pathway is the only pathway. That's a real issue because of course, lead is not the only pathway. Anybody using these tools is immediately going to say, well, of course it's not the only pathway. But the actual explanatory effect of these risk markers is less than about 5%. So when I looked I was going to present some data but didn't have time, but if we look at the overlap between ADHD, there's a recent paper now that came out from the UK Biobank looking at polygene scores that are shared by ADHD and other conditions. The thing that kind of jumped out was cognitive learning and cognitive function in adults. These are adults from the UK Biobank, so that looked like there's shared genetic markers for that. But I think the issue is we just don't have enough markers that are anywhere near the influence of the environmental risk factors. So you're right, it's such an imbalance, but on the other hand, the environmental risk factors are nonspecific. So lead could also lead to depression, it could lead to a lot of other seizures, migraine, headache. So a lot of these risk factors we look at may not be specific for ADHD. So the idea of putting it together is, I think, the biggest challenge that we have in the next decade. And you're doing a great job trying to think this through appropriately. So,
So I'd like to thank the speakers for an excellent performance of their work and the audience for their sustained attention. And I'm happy to announce the coffee break. And after that, in 30 minutes, we'll be back for the Lifetime Achievement Plenary dedicated to Marta Denkla. So be on time. Thank you.